So, good afternoon to all of you. Can you hear me? Hello? Just tell me, can you hear?
Yes, sir.
Okay,
Yes, sir.
okay, thank you. Good afternoon to all of you. It's a very difficult task for both the teacher and the student, either to engage the class immediately after the lunch or to attend the class after the lunch also. I don't know how many of you will be very attentive, and and at least I know few of you will skip or go sleep keeping your mobile on. Okay, we can't help because as far as uh, your syllabus is concerned and the schedule that has been given to me. So in the last class, we have discussed about the two important things. One is the mitochondria, or especially one thing, that is the mitochondria, and we have justified how and why the mitochondria is called as the powerhouse of cell, and various biochemical activities that is undertaken by this mitochondria helps in generating the energy which is very vital for the very survival of each and every cell in particular and as an animal as a whole. So today, in continuation with the study of the cell organelles, we will continue with another cell organ, one or two cell organelles. That is, the first one which I am going to discuss is the Golgi apparatus. The Golgi apparatus, it's also known as Golgi body, and this is the unique cell organelle which is found in most of the eukaryotic cells. Uh, Now the question is, what exactly meant by Golgi apparatus, Golgi body or Golgi complex? This is also a membrane bound organelle. Golgi complex or apparatus is also a membrane bound organelle. It is almost like a sac, and as you know, these are all the cytoplasmic inclusions in most prokaryotic cells. It's a and in absent in eukaryotes, uh, and not only in eukaryotes, even in the mammalian RBC, sperm cells of bryophytes also this Golgi apparatus is absent. This can range from one to several in a cell, and all those things depends upon the type of the cell. The in plant cell which has small Golgi complex, and that is known by dictyosome, and the main purpose and the main function of this Golgi is for transporting, modifying and packaging proteins and lipids into vesicles. So that's why this Golgi apparatus is popularly known as the packaging organelle of the cell. This Golgi apparatus has been first discovered and explained by Camillo Golgi, and for which he got Nobel Prize in 1906. So, and that's why this has been named after Camillo Golgi, and it is known as Golgi's apparatus. So, this Golgi's apparatus is made up of several stacks of parallel flattened sacs or cisternae. Each Golgi body, if you look, it is almost a saucer shaped structure, and it appears as if the saucers are piled one above the other. In addition to that, it has many peripheral tubules and vesicles coming out of it. As you can see here, see, it is a concave structure and it is just like a saucer. And it looks, there are so many Golgi bodies in it. And together all these Golgi bodies, they form the Golgi complex, or what you call the Golgi apparatus. It's a it's the Golgi structure is smooth and curvy structure. It is a flattened stack of membranes. The uniqueness of this Golgi apparatus, because of its curved nature or saucer-shaped nature, it has front end and back end. The front end is called as cis phase and the back end is called as trans phase. So naturally it has two phases. Probably here you can see. the front end, this is known as the cis phase, whereas the back end, it is known as the trans phase. So it has two phases. Front phase known as the cis phase, and back end which is known as trans phase. And Golgi apparatus has cisternae that are flattened membrane folds. Cisternae, what you call in Golgi apparatus, are nothing but flattened membranous folds. You call it as the cisternae. And along with this Golgi apparatus, you can also see number of secretory vesicles which are having many of the secretions that are to be discharged out of the cell. See the cis phase, medial region, and the trans phase. So, as I have told you, if you look into the structure of the Golgi complex, it appears as an aggregate of saucer-shaped membranes, bound structure, and each structure is called as Golgi body. In a Golgi complex, there will be number of Golgi bodies, and they appear as if each Golgi body is stacked one above the other. But if you observe this under the electron microscope, highly flattened, it appears like highly flattened membranous bag. And the space inside each Golgi body is being filled with the substances that are to be secreted out of the cell. The number of Golgi bodies in a cell is not definite. It changes. Depends on the type of the cell also, I can say that. There are, but surrounding the cell, surrounding this Golgi body, there are always innumerable spherical membrane bound vesicles which have originated from the Golgi body. Any Golgi apparatus, if you look into it, it has three zones. One in the front end, there is a cis zone facing the nucleus or endoplasmic reticulum. Because from the nucleus or from the endoplasmic reticulum, where the synthesis of the chemical going on, it receives the material to be packed. I have told you the Golgi apparatus is also known as the packaging organelle of the cell. Then comes the middle region. The third one is the trans side, that is which one is facing the cell surface. So it has three regions. And Golgi apparatus is made up of approximately 4 to 8 cisternae. So these cisternae are equally spaced stacks separated from each other by a thin layer of intercisternal cytoplasm. So as a result of this cisternae, a space is created between each Golgi body, and in between that you can see a thin layer of cytoplasm. Golgi complex has distinct polarity. The two poles are cis and trans phase. They are responsible for receiving and shipping the department. So that's why cis phase, there is the front phase which receive the things either from endoplasmic reticulum or from the nucleus. And whereas the trans phase is shipping department which send the vesicles. So that's why it is out of the cell. So that's why it is facing toward the plasma membrane. So cis phase or the convex side of the stack, then maturing phase, that is the trans phase, that is the concave side of the stack. So what happens is, if you look into that, the secretory material from the smooth endoplasmic reticulum via or through these transport vesicles reaches the Golgi complex. So Golgi, I have told you the main thing is to pack them. So for that, whatever the secretory materials, wherever they may be synthesized within the cell, that comes to the endoplasmic reticulum. So that's why while discussing the endoplasmic reticulum, I have told that it is called as the highway of the cell. Through this endoplasmic reticulum, in the form of transport vesicles reaches this Golgi complex. See, the cis side, this is the receiving side, this is the shipping side. So from here they receive the material in the form of the what you call the transport vesicles. Now small round tubules, they tubules formed from the periphery of the cisternae. There are small round tubules in the periphery of the cisternae. This is the cisternae. That is the cisternae, that is see tubules, and this cisternae form small round tubules are formed in the periphery of the cisternae. So you clearly observe, not here, actually the cisternae has formed here, see, almost a vesicle like shape. Can you make out? See, here also. So a few get enlarged at the end to form a vesicle. These tubules get transformed into or enlarges into the end to form the vesicles. Vesicles are, they lie near the end and the concave surface of the Golgi complex. So there can be different types of vesicles, smooth vesicles, coated vesicles, but all Golgi elements are filled with some fluid material. Here in the Golgi complex, you can see two networks. One is cis Golgi network and another one is a trans Golgi network. Cis Golgi network, that is the CGN, trans Golgi network, that is the TGN. They are made up of outermost cisternae at the cis and trans phase. These networks are formed, that the cis Golgi network is formed at the cis phase and trans Golgi network is formed at the trans phase. Vehicles containing proteins or any secretory material from the endoplasmic reticulum merges with cis Golgi network. See, remember, even the proteins or any secretory material coming from the endoplasmic reticulum is also in the form of transport vesicles. So this transport vesicles carrying the protein or any secretory material, they are received at the cis phase. In the cis phase, there is a cis Golgi network. So this transport vesicles merges with the cis Golgi network and thus allowing the protein to enter into Golgi complex. Now the protein that is being transported by the endoplasmic reticulum or any secretory material that is transported by the endoplasmic reticulum enters the Golgi complex. Now once the cis Golgi network receive this protein from the endoplasmic reticulum, it brings about certain modifications in the transport vesicle or the material that it has received. It begins their modification, then simultaneously moving along the membrane to membrane and slowly pushes that toward the trans Golgi network. At the other end of the Golgi complex, now newly modified protein arrives at trans Golgi network. Once this modified protein comes to this trans Golgi network, it is sent off to different parts of the cell again in the form of via transport vesicles. So this is what it does. See, from this side it receives in the form of vesicles, transport vesicle. It merges with the cis Golgi network and enters into the Golgi complex. In the Golgi body, these proteins are modified and slowly pushed toward the trans side, and that is the trans Golgi network, And there in trans-Golgi network, these modified proteins are packed in the vesicles and sent in the form of the vesicles. See here. Now, this is the general function of the Golgi complex. What are the important other functions of Golgi complex? Golgi complex has 5 important functions to carry out. One is the secretion, second one is the synthesis, third one is the sulfation, fourth one is apoptosis, fifth one is phosphorylation. These are the 5 functions. So for this, in order to understand this, probably all these functions, we have to understand this path of secretion, because the first one is the secretion. Actually, see here, this is the nuclear envelope. This is the nuclear envelope. So there will be some secretions, and that comes through this nuclear pore. And then once it comes out of the nuclear pore, this is received by rough endoplasmic reticulum or endoplasmic reticulum. From this endoplasmic reticulum as well as from the smooth endoplasmic reticulum, both as I have told. Then this ribosomes are attached to rough endoplasmic reticulum, and then in the form of macromolecules, which you can see here, macromolecules, they enter into the transport vesicles, and these transport vesicles reach the Golgi complex, cis phase of the Golgi complex, where they are modified and then they are pushed toward the trans phase of the Golgi complex. Then the trans phase pack those material in the vesicles, especially in the cisternae, and then push it out. This is one. Then the synthesis. It's a major site of the Golgi complex is the major site of carbohydrate synthesis. Even it includes the synthesis of glycosaminoglycans, GAGs, glycosaminoglycans, GAGs. Golgi attaches to the polysaccharide and protein to form proteoglycans. So this is the synthetic activity of the Golgi. Golgi is a major site of carbohydrate synthesis. It can attach to polysaccharides and proteins to form proteoglycans. Another function of the Golgi complex is sulfation. Golgi involves in the sulfation of certain molecules. That is adding a sulfur group, sulfation. Passing through certain molecule, passing through the lumen via sulfotransferases that gain sulfur molecule from the donor called PAPS. Okay. Golgi involves in the sulfation of sulfur molecules, certain molecules. Certain molecules require a sulfate group. So for this, what the Golgi does is, when passing through the lumen via sulfotransferases, that gain a sulfur molecule from the donors called as PAPS. Then that sulfur molecule is generally performed, sulfation is generally performed at the trans-Golgi network or TGN. The sulfation can occur at GAGs of proteoglycans as well as the core protein. This sulfation is very essential, very important to proteoglycans, which is a synthetic activity. Now, it adds a group of sulfate. Then the third one is apoptosis. What is apoptosis? Apoptosis is called as, in simple, it is a programmed cell death. It's a programmed cell death. After the certain lifespan, each cell will die. Each cell has a definite lifespan. For example, our RBC has lifespan of 120 days. Like that, each cell has a. So this Golgi apparatus has a role in apoptosis. A newly characterized protein that is known as Golgi anti-apoptotic protein. It's a newly synthesized protein. That is the Golgi anti-apoptotic protein that exclusively lies in the Golgi and protect the cell from apoptosis. So actually the cell death is prevented by this Golgi anti-apoptotic protein, which remains in the Golgi and it protects the cell from apoptosis. Otherwise, the premature death of the cells can happen. But the process is not very clear yet, and because how it does, it's we can only say that by some undefined mechanism it does. Now, transport, vesicular transport. What actually here does, as we have already explained, it's a process in which membrane enclosed transport vesicles. When there is a transport vesicles, they are membrane bound, in which the maybe transport proteins, most of the time it is the transport proteins, from one membrane enclosed compartment to another membrane enclosed compartment. Because these proteins are also enclosed in the membrane, even Golgi bodies are also enclosed in the membrane. This protein doesn't move across the lipid bilayer. The protein which is found in the vesicle, transport vesicle, doesn't move across the lipid bilayer. Instead, only move between the compartments which are topologically equivalent, like the lumen of endoplasmic reticulum to lumen of Golgi. They can, because both of them are topologically equivalent compartments. They can move. They cannot cross the membrane. So now, this Golgi apparatus transports protein, lipids and polysaccharide to their final destination. That is what you call secretory pathway. What it has to do is, there should be sorting of protein into different kinds of transport vesicles. Golgi complex receives number of transport vesicles coming from endoplasmic reticulum. Now, once it receives this transport vesicle, the function of the Golgi complex is to sorting different kinds of transport vesicles. Then, after sorting, you bundle them. And then the trans-Golgi network, through the trans-Golgi network, they are again packed and deliver their content to the appropriate cellular location. This is what is done in the. So here, transport of the Golgi complex, which you can see here photographically, one is constitutive secretory pathway and another one is regulated secretory pathway. There are two pathways for the transport from the Golgi apparatus. One is constitutive and another one is regulated. In constitutive secretory pathway, proteins are secreted from the cell continuously. The proteins are secreted from the cell continuously regardless of the external signal or factor. Continuously there will be production, secretion. Now what to do with this protein? These proteins are stored in vesicles in the Golgi, and then they are moved directly to the cell surface, where it fuses with the plasma membrane and then release the soluble protein outside. See, this is the step where there is one, the Golgi vesicles. From here they move to the cell surface, that is the plasma membrane. They bind with this plasma membrane and release the content outside. This is in the first one, constitutive secretory pathway. But if you come to regulated secretory pathway, here the cell does not go on secreting the protein continuously. Instead of that, it produces some specific proteins, only specific proteins, in response to the signals, in response to the environmental signals. Now, these produces specialized or distinct proteins are sorted in the trans-Golgi network and packed into secretory vesicle. These secretory vesicles are usually larger than the transport vesicles. Transport vesicles are smaller, whereas the secretory vesicle that has been produced by the Golgi complex is always larger or usually much larger. And they are stored within the Golgi complex until they receive specific signals. Only after receiving the specific signals, they are released from the Golgi complex, fused with the plasma membrane and release the protein. Till then, they are stored in the Golgi complex. Now, how the sorting of the protein can happen in the Golgi process? The process of protein sorting in the Golgi, it is through lysosomes, through selective transport of proteins, we can say that. The Golgi apparatus, along with the lysosomes, has the capacity to identify them. Then the proteins are modified into mannose phosphorylation by the process, the proteins are modified by a process known as mannose phosphorylation in the cis Golgi network. Now, this modified, that is the phosphorylated mannose molecules, are specifically recognized by mannose 6-phosphate receptors in the trans-Golgi network. This is very interesting how it is able to sort. Once the protein comes to the cis side, these proteins are modified by mannose phosphorylation process. Once these proteins are modified, that is phosphorylated, now they become phosphorylated mannose molecules. This phosphorylated mannose molecules can be detected by some detectors which are on the surface of the trans-Golgi network, and those receptors are known as mannose 6-phosphate receptors. So this is how actual sorting takes place here. Now, I have told you in the Golgi complex there are two phases, cis phase and trans phase. In between there is a middle region also. Cis phase originates from smooth endoplasmic reticulum and it collects the secretory material in the form of transport vesicles from the smooth endoplasmic reticulum. Actually, it is in the middle region, whatever the modification that takes place to the secretory substances. Then it is pushed to the trans side where actively those substances are packaged into Golgi vesicles and they are sent out through the cell surface. So that's one is the first, the modification of the lipid and protein, then storage and packing of the material that will be exported from the cell. See, this is how the same photograph which you have seen already. See, these are the It has come from endoplasmic reticulum in the form of transport vesicles received by cyst side. In the middle region and trans side they are modified, and in the trans side they are sent out as a form of Golgi vesicles. And that vesicle fuses with the plasma membrane and pushes the material outside the cell. So that's why it is also called a shipping department. So the vesicles that pinch off from the Golgi apparatus move to the cell membrane, and the materials within the vesicle are released to the outside of the cell. The very interestingly, some of these pinched off vesicles also become lysosomes. Among these protein modifications, Golgi apparatus, as I have told you, are involved in the transport of lipid around the cell. Sorting, these are all the things which we have discussed. So this is the photograph of an electron microscope. This is the trans side and cis side, and you can see numerous vesicles. So this is how it does. See, I hope you can see this small animation. See, it receives, and the Golgi apparatus, what it throws is the Golgi vesicles containing the proteins. See, from it, the cis side it receives, and vesicles are pinched. Can you see? ಕಾಣ್ತಾ ಇದೆಯಾ ನಿಮಗೆ? Hello?
Yes, sir.
Okay. This is the mechanism how the Golgi complex works. So, even Golgi complex is found to play an important role in Alzheimer's disease also. Alzheimer's disease is a brain disorder where brain cells are being destroyed. It has been observed that death of the neuron affects the brain and there will be shrinking of brain. You can see here, this is the perfect healthy brain, this is the advanced Alzheimer, how the brain shrinks. It has been observed that Golgi apparatus, if it is not functioning and it is not carrying out the process of collection, transport and distributing the protein molecule, the Alzheimer's disease can happen. So, stacks of sacs and vesicle pinched off from the sacs. So, this is about the Golgi complex. I hope all of you have understood about this. Then, let me move on to the next organelle, that the lysosomes. These lysosomes are also equally important. Very, very interesting. Undoubtedly, these lysosomes are also membrane-bound organelles, and they are found in most of the animal cells. They appear as small membrane-bound inclusions in the cytoplasm. Actually, many times it goes very difficult to differentiate between vesicles and lysosomes because both of them are membrane-filled inclusions. Membrane-bound inclusion in the cytoplasm. The number of lysosomes are highly variable in a cell. Suppose with the light microscope you will not be able to see lysosomes, but when you observe that under the electron microscope, it appears like a small circular area filled with some dark components. Small circular area, because it's a vesicle that is being filled with some dark substances. This lysosome is rich in hydrolytic enzymes. That is the enzyme which can break down, break down. It is rich in hydrolytic enzymes. So these lysosomes are popularly called as digestive plants for protein, fat and carbohydrate. Generally our food contains only these three: protein, fats and carbohydrate. So that's why The lysosomes are popularly known as digestive plants. It is also very essential for the transport of undigested material. Undigested material for the removal. For that, this undigested material should be pushed through the plasma membrane outside. Suppose if the lysosomes explode within the cell, the entire cell bursts, break down. If the cell breaks down, if the lysosomes explode. So that's why the lysosomes are known as suicide bag of the cell. Suppose if lysosome bursts, releasing the hydrolytic content in the cytoplasm, the entire cell gets burst, dissolves. So that's why if the cell want to commit suicide, it can activate the lysosomes and make the lysosomes to burst, and there will be a total death of the cell. Actually, lysosomes are not discovered by electron microscope, but they were found by centrifugation and enzyme analysis that has been identified. It's simple, spherical and single membrane bound structure. Simple, spherical, single membrane bound structure. Lysosomes contain a large number of catabolic enzymes, remember. In enzyme there are two types, anabolic and catabolic. Anabolic enzymes are the ones which are constructive. Catabolic ones are which are meant for breaking down. So these lysosomes contain large number of catabolic enzymes. And the role of the catabolic enzyme is to digest the material by the process of hydrolysis. See here, this is the anatomy of a lysosome. That's a membrane, plasma membrane, inside which you can see the hydrolytic enzyme mixture. And outside, that's a glycosylated membrane transport proteins here. And this is very similar to that of the plasma membrane, which is having a lipid bilayer. What are the enzymes that are found in the lysosomes? All are hydrolytic enzymes, remember. One is acid phosphatase, which acts on phosphate esters and breaks it down. Even acid ribonuclease, which acts on RNA and digests it. Acid deoxyribonuclease, it acts on DNA and hydrolyzes it. Glycosidases, these are the hydrolytic enzyme which can act upon polysaccharides. Proteases can act on proteins and peptides. Lipases can hydrolyze the lipids, and phospholipases acts on phospholipids. The entire cell is made up of these substrate only, and there is a corresponding enzyme to hydrolyze it, and all of them are found in the lysosomes. So that's why I have told you lysosomes are very, very rich in hydrolytic enzymes, and they contain all kinds of hydrolytic enzymes. In fact, these are some of the important hydrolytic enzymes. There are more than 40 different enzymes are known to occur in the lysosomes. Enzymes are capable, so that's why it is called a suicide bag of the cell, bags of the cell. Enzymes are capable of breaking down or digesting almost any biological material. Because now we have seen the list of the hydrolytic enzymes. They are capable of breaking down or digesting almost any biological material. But only thing, it is bounded by a specialized membrane. These hydrolytic enzymes are bounded by a specialized membrane which doesn't allow this hydrolytic contents to go out. and they are keep the content of the lysosome inactive, and that membrane keep the hydrolytic enzymes inactive, but at the same time ready to use. So these lysosomes are usually meant for intracellular breakdown. Intracellular, within the cell, the breakdown of all the biological material. Okay, now the question comes, when this happens, why the cell wants to commit suicide? Why the cell wants to die? Or when it happens? There are many instances when the cell dies. But don't think every time the lysosomes are used for committing suicide. No. There are, for example, the phagocytosis. In general, all of you know it is the cell eating process. Suppose when solid bodies like bacteria or other germs are ingested, when they get ingested, they are engulfed inside a small food vacuole. This food vacuole is brought to the vicinity of the lysosome, is brought closer to the lysosomes. And then this food vacuole lysosome, they merges into one. When the food vacuole and the lysosome fuse together, this complex structure is known as secondary lysosome, which has been formed by the fusion of lysosome and the food vacuole. Now, what happens? There are many digestive enzymes inside the lysosome, which all of you know. These digestive enzymes acts on the food vacuoles, and its contents are broken up and digested by the content of the lysosome. Thus it protects the body. Suppose any bacteria or any of the pathogen enters, first they are coated with a vacuole, and that is called as the food vacuole. And this food vacuole is brought to the lysosome. Lysosome merges with that and forms secondary lysosome. Then the digestive enzyme within the lysosome digests the entire food vacuole, and contents are broken up, hydrolyzed, and thus protect the cell. See, this is the lysosome, this is the food vacuole, together it is called as secondary lysosomes, and inside this food vacuole, whatever the bacteria or pathogen, it is being digested, and thus it protects the cell. See here also. It binds with the foreign particles and digests. In lower animals like Protozoa, Coelenterata and Porifera, where the intercellular digestion takes place, intracellular digestion, this digestion of the food is with the help of these lysosomes. What happens if an amoeba engulfs the food material? Then this food material is packed with the help of a vesicle and called a food vacuole, and that food vacuole merges with the lysosome to form the secondary lysosome, and there the digestion of the food occurs within the secondary lysosome, and digested food is then absorbed. So, in fact, you can say that it's a means of what? It's a means of digestion in lower organism. In higher organism, it's a means of protection. Autolysis, self-destruction. The content of the lysosomes, as I have told you, are capable of digesting or breaking down almost all components of the cell in which the lysosomes are formed. If this happens, the process is known as autolysis. What do you mean by that? The, so, if the cell has already been infected and if the cell is able to digest the pathogen, then what happens? It will kill the entire cell by activating and thus releasing the hydrolytic content of the lysosome. This is so that's why it is known as suicide bags of the cell. Or sometimes old age, the cells are being digested by this particular process, what you call autolysis. Or if some cells are attained their maturity and they are no longer needed for the, then also this cell can activate the lysosomes. How? By releasing its content into the cytoplasm, and once the content, that means the hydrolytic enzyme, reaches the cytoplasm, virtually they can kill the entire cell and get assimilated. This is what happens, how the lysosome works or functions. So for the day we will stop here, because even if I start centrosomes, I will not be able to. Probably in the next class, we will, tomorrow I think, we will complete this cell organelles and move to the what you call the nucleus. So was it clear? Really? Srinidhi? ಹಲೋ ಶ್ರೀನಿಧಿ,
Yes, sir.
ಕೇಳ್ತಾ ಆಯ್ತಾ? ಸರಿ ಅರ್ಥ ಆಯ್ತಾ?
Yes, sir.
Could you
Great.
understand the Golgi complex and this both Golgi complex and lysosomes?
Yes, sir.
Madhu,
Yes, sir.
any doubt?
No, sir.
Uh, so in that case, I will stop here, because I will not be able to complete the discussion on the centrosome. So that's why tomorrow I will take it up. Any doubt? Anything else? Anything to be asked? Still we have 1 or 2 minutes left. Yes? Ready? Yes? If not, we will stop here and we will meet in the next class.